Maxam and Gilbert sequencing. This method is also known as the chemical method. It involves determining the nucleotide sequence of a DNA that is terminally labeled by using chemicals that break the DNA chain at adenine, guanine, cytosine, and thymine, specifically.
This creates N-radioactive labeled fragments that together with a polyacrylamide gel electrophoresis can be identified by size. Revealing these points of breakage in order, it is important to note the reaction will take place separately, so that results by 4 autoradiographs that will show a pattern that will be identified and sequenced. The DNA strands are degraded by heat. Sequences of DNA labeled with 32-phosphate at the end will hybridize with the cut strand, creating radio-labeled strands. Electrophoresis will reveal the different size strands. Different chemicals change the composition of nitrogenous base at specific positions. This change will weaken the bonds between the bases and the sugars, creating a break. In purines, adenine and guanine, dimethyl sulfate will attack by methylating a nitrogen atom, making the bond unstable that can be broken by increasing heat. An alkali solution will break the sugar from the phosphate group. Brightest band in the electrophoresis will indicate when guanines were broken, and light bands will tell when adenines were broken.
With the addition of dilute acids, adenine bonds will break easier, giving more adenine breaks. This means that brighter bands in electrophoresis will correspond to adenine breaks, and the lighter ones to guanine breaks. In pyrimidines, cytosine and thymine, hydrazine is used to break the strand. The base changes to ribosylurea, and hydrazine keeps reacting until a solution of pyrimidines cuts the molecule, the sugars, and the products of the previous reaction. The bands in electrophoresis of cytosine and thymine cuts are similar. When sodium chloride is used, it suppresses the effect of hydrazine in the thymine bases, so the resulting bands are only of cytosine. After analyzing each trial, the sequence can be deduced.
DNA contains nitrogenous bases, phosphates, and carbohydrates. They can be classified according to their spatial arrangement as D or L. DNA is made from D-deoxyribose. An area of opportunity in biomedicine is the creation of L-DNA aptamers, which are a single-stranded oligonucleotide that can target specific biomolecules. These oligonucleotides have medical uses, especially the creation of target-specific drugs. They have not been used because they are easily degraded by nucleases in the body, specifically in the blood.
LDNA, as its reverse image, cannot be degraded by nucleases. Aptamers using LDNA could have the potential to create protein-specific drugs, but the sequencing of LDNA is very hard because the current methods do not use chiral reagents. Leo and Sue have been using Maxam and Gilbert sequencing method, which uses a chiral reagents that allow them to create DNA. This has allowed them to sequence LDNA oligonucleotides of 55 bases long, holding the promise for many exciting applications in biomedicine.